Get ready to enter a brave new world with your host, Vasant Dar.
Brave New World is supported by the Center for Data Science, or CDS, at NYU. If your organization is interested in engaging with CDS through student projects, please email cdsindustry@nyu.edu. For more color on the podcast and additional commentary, please subscribe to my newsletter at vasantdar.substack.com.
Hello, and welcome to Brave New World. My guest today is Sandeep Robert Datta, who heads up the Datta Lab at the Harvard Medical School. Sandeep is a pioneer in the study of smell and the brain, and his research explores the link between chemistry, perception, and behavior. Sandeep, welcome to Brave New World. I am delighted to have you on the show.
Well, thank you so much for having me.
Before we sort of launch into the meat of the session, just tell us a little bit about yourself. What influenced you growing up, and how you ended up at the Datta Lab in Harvard, running the lab.
Yeah, sure. So when I was growing up in high school, I was super interested in science, but I never thought of myself as a scientist. And so when I went to college, I thought that I'd become some sort of banker. And then I finally took a bunch of classes in economics, which I realized that I really hated. But I was really loving the basic science classes I was taking. I remember as a freshman, I took organic chemistry, which was just amazing in terms of its ability to kind of describe the world and make predictions about how molecules came together. And I was just enchanted. And so I started taking more and more science classes. And I ended up my sophomore year joining a lab studying fruit fly development. We were interested in how the fruit fly gets its various appendages. And we thought about how, genes and individual cells might participate in that process. And as I did more and more basic science, I realized it was just a animating force in my life. I just loved it. And so, like a dutiful Indian son, I applied to medical school and was admitted. And my mother could not have been happier. And then when I got here to Harvard, I was admitted to the MD program. While I was here, actually, I decided that I really wanted to do a PhD in basic science. And so I took a left turn and I joined the doctoral program here, where I worked with Dr. Michael Greenberg to study signal transduction, the process through which cells understand what's going on in the outside world. And I loved that so much that I decided after my MD not to become what my mother would refer to as a proper doctor. So she is, of course, disappointed to this day. But I then decamped for Columbia, where I worked with this guy named Richard Axel, who was a giant in the field of molecular neurobiology. He's famous for having cloned all sorts of really critical receptors. He cloned the receptors for HIV. He cloned CD4 and CD8 in the immune system. He identified the receptors for glutamate, for example, which is a key neurotransmitter in the brain. And he ultimately won the Nobel Prize for discovering the receptors in our nose that allow us to smell, the odor receptors. And I was actually in the lab when he was awarded that prize. It was just a tremendously exciting time. While I was in the lab, we began moving from thinking about how the nose itself detects odors to thinking about how the brain might process odors. And so I did basic work in the fruit fly and the mouse, trying to understand the underlying structure of the olfactory system to give us some ideas about how the higher brain might make sense of smells from the outside world. And then in 2010, I came here to Harvard, where I started my own lab, and I focused on very similar problems.
So how did you decide to focus on smell, when you were thinking about what to do? What is it about smell that was especially appealing to you? Was it because you were working with Axel, or did it develop before that?
In truth, the reason why I'm interested in smell is because smell is one of the few examples in the nervous system where there's a clear relationship between the function of individual molecules and patterns of activity that are going on in the brain. And so, we have these odor receptors that detect odors in the nose, and those odor receptors don't just detect odors, they actually define the anatomy of the olfactory system in the brain. And that anatomy, in turn, defines the ways in which the brain interprets smells in the outside world. And so if you're interested in how, genes might lead to the generation of molecules that ultimately might pattern the nervous system and give it its function, the olfactory system is it's like an ideal setting to think about that very basic problem.
There's a lot in there. And, I'm reminded of, you probably heard of Joy Milne, who found that she had the ability to smell Parkinson's and now other diseases as well. So you said something a couple of minutes ago, and I'm going to try and paraphrase it. You said something like that the olfactory receptors in our nose somehow map on to something in the brain. Tell us a little bit more about that. Well, what is the relationship?
I'm happy to talk about that. It's really interesting, and it's actually why many people who are interested in smell study it. So in your genome and in my genome, there are about 400 separate genes that encode odor receptors. In the mouse, there are about 1,000 of these genes. And in dogs, there are about 2,000 of these genes. These genes take up more genomic real estate than any other gene family in the mammal. So it just tells you how important smell is.
What do you mean by that when you say they take up more genomic real estate than other senses?
Than any other gene family, actually. So there are more genes for smell than genes of any other type for any other function in any cell in your body. So like the most genes of a given type that you have are odor receptors. So they're the single largest gene family in mammals. So they occupy a huge amount of your genome because there's so many of these genes. And so from an evolutionary perspective, the fact that the genome has devoted so much of its resources to genes that code these receptors kind of tells you how for most animals, smell is so very important.
It's ironic that smell accounts for so much of, the genomic real estate, as you call it. And yet it's one of the least appreciated sort of senses. In a sense, people sort of shy away from smell. Because I'm a big smell person myself. I like to smell things. Yeah. And, and I remember we see, at least in the United States, we seem to have like some sort of a cultural aversion to putting a nose next to something.
Yes.
It's considered, I don't know, crude, crass, whatever.
Impolite. That's right.
Sorry?
It's considered impolite. Just kind of sniffing around, it seems.
That's right.
Yeah.
That's right. And so when I first came to the US and I was not sufficiently culturally acculturated, I used to go around sniffing things a lot more. I just am attracted to smells, and people would look at me strangely. And so I feel it's sort of ironic that here's this sense that seems to have played such a large role in evolution, and yet we seem to sort of run away from that. What's going on here?
Yeah, so, for most animals that aren't in our primate lineage specifically, smell is arguably the most important sense they use to engage with the world. So whether you're a fruit fly or a mouse or an armadillo or even arguably a bacterium, the way that you interact with the world largely is through chemical sensing, by detecting small molecules that are out in the world and understanding what they are. We, of course, have this just spectacular visual system, right, that allows us to see in color and 3D. And as we evolved as visual creatures, relative importance of our sense of smell and our dependence on our sense of smell for things like navigation have diminished. That being said, we're still possessed of a truly spectacular sense of smell as humans, and one that, for evolutionary reasons, still plays a kind of critical role in our sense of well-being and our sense of memory, and it centers our emotions. It's even though we don't think of it as being particularly important, I think, to us as humans, it's still this really essential part of who we are. That said, because we've acquired this really robust visual sense, our dependence on smell is less than in other animals. And I think for those reasons, and because, we communicate through language, which we hear. For those reasons, when neuroscience was invented, actually by folks who were working just a floor away from me, the first recordings they did were of cells, that responded to visual cues. Like the things that people were excited about and were about understanding, in the 1950s, how individual cells in visual cortex responded to dots or to lines. And so historically, we've paid a lot of attention to vision, and we've paid a lot of attention to audition and far less attention to smell, even though for most animals, smell is a much more important sense than vision.
I've been, I've really sort of gotten into smell the last couple of years, initially just out of curiosity, but now as a researcher. And, as I've been sort of reading up on it, I'm finding assertions or maybe people pointing to evidence that many times, when we develop diseases, for example, we oftentimes lose our sense of smell first, and we're not even aware that we've lost that sense. How much do we know about this? Because I can imagine it's hard to study. So how much do we know about this?
Very little, actually. So here's a basic question whose answer we don't know. Basic question. After you get a common cold and you get better, what fraction of people still have a loss of their sense of smell? Right? It's a basic question you'd imagine we'd know the answer to. That basic question we don't know. And so when everyone started losing their sense of smell after they got infected with COVID, kind of as a community, we didn't have a whole lot of answers because we haven't really studied and we don't really understand the underlying pathophysiology that just in general, for any cause, leads to a loss of the sense of smell. Right. So in no settings, for example, are there treatments for the loss of the sense of smell? Like if you get a common cold, which can sometimes cause a prolonged loss of sense of smell, we don't know what to do. If you're going to go on to get Alzheimer's or Parkinson's disease, often in kind of late middle age, people begin to lose their sense of smell, and that can be a sign that you're going to go on to have a cognitive disorder, right? But in those people, often, as you were mentioning, they don't notice. And even when they do notice, there's nothing we can do. We don't know what to do. So it's just a very understudied space. And one of the consequences of that actually is that we just know very little about kind of basic principles, and our thinking is dominated by little anecdotes. So another question you might ask, to which we don't know the answer, is how many smells can a human smell? How many smells can a human tell apart? Right? That's also a question whose answer we don't know. Is it, the answers in the literature range from, on the low end, 10,000 to on the high end, 1 trillion. And what the actual answer is, no one knows.
Like, what does that question even mean? Like, how many things we can smell? Because it seems to me that even I guess my question is like, is that question even answerable? And the reason I ask that question is because for the most part, we rely on language for everything, right? We rely on language to describe things, and vision is great because we can describe a tree, a lamp, a car, whatever, and all agree that we're seeing the same thing, right? And most of the time we do see the same thing. There can be times when things are ambiguous, but vision to me has this sort of very firm notion of truth that we can all agree on, right? Whereas, when I really start getting into smell, it seems that when we try and describe smell in terms of language, I don't know, it just seems inadequate. It doesn't seem to describe the richness of smell, right? So and what I really want to get into here is more, the notion of meaning, right? What does smell really mean?
Yeah.
Right? How do we start ascribing meaning to odors?
Right. So let me just kind of discuss for a moment the first thing you mentioned, which is, as humans, how can we get some sense of what it is we are smelling and talk to each other about it? So, one strategy that of course one can use is just natural language. I give you a vial of something, you and I are hanging out at Saks and we're smelling the same perfume, right? And we have a conversation about what it is we're smelling, right? And so, I might use a bunch of terms like, oh, it smells like lavender and jasmine and peppermint, so on and so forth. And you might smell and you might agree or disagree. And that's, us using descriptors and essentially analogies to try to capture qualitatively and what we're experiencing subjectively. There's another way that we as humans can communicate about what we're smelling. And that is by talking about essentially relationships. So I think you and I would agree, even though we're separated by a great distance and we're not in the same room and we're not smelling the same thing, you and I would agree that lemon and orange are more similar than pizza to each other than to pizza, right? Like lemon and orange are they're somehow grouped together qualitatively into some sort of category. And so they have some sort of relationship with each other that's distinct from the relationship that either of those citrus fruits has with pizza. I think we would agree, right? And so one can kind of get beyond language by performing tests on humans where you just ask humans, you give them two things to smell, then you give them a third thing to smell and you say, all right, match them. Which are the closer pair? And by doing that, you can build a kind of map. You can identify smells that humans think are more closely related and identify smells that humans think are more distantly related. And it turns out that those kinds of maps are at least in part shared across humans. And so there is a kind of commonality in the ways in which we perceive odors that we can identify explicitly, because this kind of relational map of smells that's in your nose and in your brain is at least somewhat similar to mine. And we know that again because we both agree that lemon and orange are similar to each other and they're both different from pizza.
Yeah, that makes a lot of sense. And it establishes that there's a quote unquote distance measure between how we perceive different molecules or mixtures of molecules in our brain. To what extent is such a distance measure cultural or genetic or environmental, that is based on the smells that we grow up with? And the reason I ask this is because, I remember, when I was 19, I hitchhiked from Kabul to London, and on the way in Switzerland, stayed with a friend who decided she was going to make us fondue. And we went into this cheese shop and I almost passed out. I mean, I could not take the smell. I mean, to me, it was, I hate to use the word because I know there's lots of cheese lovers who are listening to the show. But to me, it was just like, anything but pleasant. And yet I know, there's a tribe in Corsica or Sardinia, I don't recall, who relishes this byproduct of maggots, processing, milk and producing this thing. And so I'm thinking we agree citrus and orange, and that seems reasonable, but there are these other things that just seem so dependent on culture and where we grew up, and maybe that affected how our brain got wired.
Let me ask you a question. How do you feel about cheese now?
Much better.
Yeah, right. So what does that tell you?
Well, yeah, exactly. So I have become acculturated,
yep.
but I'm still not a big blue cheese fan, right? So I stick to the safe cheeses.
Yes.
So you're exactly right, though. I mean,
yeah. I mean, what this tells you, right, is that it's probably true that to some extent the way in which our brain processes smells is deeply experience dependent. And it's probably true that early life experience plays an outsized role in shaping that kind of processing. I think you should know is that the parts of your brain that are responsible for smell are probably the most flexible and plastic parts of your brain. So a lot of people like me think a lot about how your higher brain processes smells, not just because we're interested in smell, but because that part of the brain is actually directly connected with the key part of your brain that's involved in learning and memory, which is called the hippocampus. It shares an architecture with the hippocampus. The cell types that live in this part of your brain, which is called the piriform cortex, look very much like the cell types that live in hippocampus. And they actually are interconnected, and they co-evolved, actually. They both evolved at the same time. So the part of your brain that's responsible for memory co-evolved with the part of your brain that evolved for smell. And all of that evolved before your sense of sight or your sense of hearing, believe it or not, which is why, I refer to olfaction as being such an evolutionarily ancient sense. And what this means is that, the olfactory system, the higher centers involved in olfaction, they're really flexible. They're constantly learning about new odor relationships and adapting themselves to your experience. And they're also very sensitive to, your reactions to smells and your interactions with smells. And so you can quickly reassign a smell from being pleasant to unpleasant, as you have done with most, but not all Cheez-Its in the course of your lifetime, right? The olfactory system is remarkable at learning associations between smells and things that are pleasant or unpleasant or rewarding or punishing in different ways.
Yeah, very, very interesting. You mentioned memory, and I want to come back to that because one of my previous guests, Michael Levin, I had him on the show a few weeks ago, and we were talking about memory, and we take memory as something that's like passive. It's stuff stored and recorded, and he was talking about how memory is actually created, which is a very different sort of way of looking at how we store and retrieve information. I'm going to tell you a little story and ask you to interpret this. So a friend of mine was in New York City at the US Open. He went back to his hotel, got up in the morning and went for a run. And he had a cardiac arrest on 42nd Street and 7th Avenue. Apparently, there was an Israeli tourist woman passing by who gave him CPR, and there was an ambulance that happened to be passing by there. This was on a Thursday morning, took him to the NYU Medical Center, and he resurfaced on Monday night, and people had no idea whether he still had any kind of brain function. And one of the things he recounted was that he has no memory of the US Open. That all he knows is that he went there, but he has like zero recollection of the match that he saw, which I thought was like so bizarre and interesting, right? Because, you would think that you're watching the match and this data is getting sort of imprinted in your brain in terms of memory. And yet it isn't, and then, when he came to, they asked him, what year is it? And he said, I don't know, 2022. And then he talked about how even knowing what date it is sort of constant reinforcement. We keep looking at the date around us. Like, what date is, it's October. Oh, it's already November. Actually, it's November 2024. So it made me think that like memory is almost like, number one, it takes time to get established in the brain. And, I hadn't actually thought about that. I mean, I could buy that we sort of retrieve, that we store the essence of something. And then when we retrieve it, it may not be the same thing. It'd be maybe, reconstructing events and stuff like that. But I had never actually thought about the fact that the US Open wasn't even recorded.
Yeah. So, I think classically we think of memory as evolving over different timescales. And memories is being passed from kind of memory system to memory system as you consolidate memories over time, such that, as you were mentioning, it takes time to really kind of permanently store something and remember something. So for example, if I were to give you, 4 numbers to remember in sequence, and I would ask you, 30 seconds later what those numbers were, you'd easily be able to do that. And that would be true because you have something called working memory that allows you over relatively short periods of time that are measured in seconds, to remember things. There's also forms of memory called short-term memory that depend on a different part of the brain, actually, that are about the kind of initial gathering of information and crystallization of that information into a kind of memory. And then there's forms of long-term memory where, short-term memory systems pass that information forward to other neural systems that store memory for longer. So memory is not like one thing; it's many, many layered things, and it can reflect experiences that evolve on many different timescales. What's interesting?
Do we have any kind of idea as to what these things actually look like biologically? Right? Because, I grew up with sort of, Herbert Simon, and, he had these sort of models of thought and, in terms of long-term memory, short-term memory, and there's a noticing and evoking mechanism, right? But it was sort of an abstract computational description of memory. What is this thing? Do we have any idea what this actually looks like in the
Yeah.
brain, like the short term, long term? Like what's actually going on there at the biological level?
Yeah, I think we're beginning to have some sense. I will say that there's still very basic debates over what constitutes a memory and how a memory might be stored. So one idea, which is an idea for which there's evidence, is that the fundamental unit of memory is a neuron. And so memories are stored in groups of neurons called engram neurons. And if you somehow are able to artificially kind of tap on these engram neurons and activate them, you can, in an animal at least, elicit a memory. So that's one idea, right? And it seems like, at least in certain contexts, that is true. So you can, for example, associate a shock with a particular environmental context in an animal. And then you can identify neurons that are getting activated as a consequence of that association. And if you then use fancy neuroscience techniques like we use in the lab to reach into the living mouse's brain and activate just those neurons that were activated during the formation of the memory, you can actually evoke a behavior in the animal as if it's getting shocked, even though it's not, if it's about to get shocked, even though it's not. So that idea is the idea that the kind of fundamental unit is a population of neurons in your brain, somewhere in your brain, that's storing the memory. Another idea is that actually the fundamental unit is the connections between neurons, the synapses, and that the information is fundamentally stored not in static populations of neurons, but in the connectivity that interconnects neurons and therefore interrelates different parts of the brain and allows information to pass through the brain in various ways. And as a field, we're still debating whether either or both of these models is true. I think most people think both ideas have some truth to them. That, we know that as a consequence of experience, connections between neurons can be shaped and reshaped. And so an essential part of learning is reshaping how neurons talk to each other. And we also know that, as I mentioned, that it seems like individual neurons are important for storing and recalling particular memories. So it's probably some combination of, experience somehow rewiring the ways that neurons talk to each other and memories being specifically stored in subsets of neurons. But the details of this are still, I would say, super vague for even for those of us in the field.
So let's drill down a little bit more, because it seems to me that the details here will really matter. So how are you actually studying these details in your lab? Because when I went into your description, you said the main goal of the Datta lab is to reveal how the brain composes natural behaviors that are endowed with purpose. Now let's connect this to the actual sort of dirty work you do in the lab with mice, getting inside their brains and examining them. Say a little bit more about sort of the methods and the data that you use in order to sort of get into these details.
Yeah, so we look at kind of two main domains in thinking about how the brain works. So the first is we think very carefully about how information, specifically about smells, is detected by the nose and then processed, stored, recalled by higher order regions of the brain. And we think about how these sensory regions that are responsible for processing information about smells, we think about how they might be connected to centers in the brain involved in emotion, memory, and especially behavior. So that's like half of what we do. The other half of what we do is to think very seriously about behavior itself, to ask, why animals, in the case of my lab, I'm talking specifically about mice, why mice in a given circumstance behave precisely the way they do at any given moment in time, and how their patterns of behavior might be shaped by what they're sensing in the world, which in the case of our lab is nearly always different kinds of smells. And I want to remind you, the way that we interact with smells is through sniffing, as you were mentioning at the very beginning of this. And so detecting smells and behaving are fundamentally intertwined, because you have to
and by behaving and by behaving you mean what? Like what kinds of behaviors are you observing?
I mean both the overall movements of the animals and movements of the animals that mice generate in order to better sense the world, which are generally referred to as active sensing. a sniff where you volitionally move your nose and your lungs and you take a sniff. That's a thing that your motor system, your behavioral systems generated in order to give you more information about the sensory world. And so there are behaviors you can generate, right? Like going up to the cheese counter and taking a big, deep sniff that are under your control and volitional and designed to help your sensory systems understand what's out there. And then in turn, your sensory systems give you information, and that informs your next behavior. So imagine you in your fondue shop. You go in, you take a big active sensing movement, you take a big sniff in, you smell this horrible smell, and that tells your nose to stop sniffing and your body to recoil backwards, to get away from the smell, right? So sensation and action are fundamentally interrelated in these loops, which we in neurobiology refer to as sensory motor loops, in which one is constantly informing the other and the other is constantly informing the one. And so you can't really tease apart the sensation part and the action part. They're really intertwined. And so I think about the brain as working through these loops. And so we think both about kind of how smells are detected and encoded, and then how that information informs movement, and then how movements help you with smell. So we try to do experiments that go all the way around that loop.
So give us an example of an experiment like this.
Sure. So a basic experiment that we would do, just to give you a sense of it, would be to have a mouse run around in an empty bucket. Okay. And we'll watch the animal explore the bucket. And we'll use methods that we have developed over the past decade in the lab that are machine learning based, that allow us to essentially decode the body language of the mouse. So we can, when we as humans watch a mouse running around in the bucket, it looks like it's just kind of, fidgeting a little bit and darting one way and then running another way, then stopping and sniffing and turning and running along the walls and occasionally running across the center. And just as is true for smells, we as humans have very poor language for describing what a mouse does. We just don't have the language for it. But through, careful machine learning based analysis, we can actually discover what we call the behavioral syllables out of which the mouse's behavior is actually organized, and describe the behavioral grammar that governs the order in which the animal generates different movements. So it turns out that mouse behavior in a bucket, even though to us as humans it may seem pretty random, is actually highly structured and predictable and determined by the animal's genome and the context you put the animal in. And so we would do an experiment where we take a mouse in an empty bucket, we characterize its behavior, and then we introduce a smell into one corner of the bucket, right? And we watch the animal's body language change as it interacts with the smell. And then often in these experiments, we'll simultaneously have a silicon probe implanted somewhere in the animal's brain, maybe in an olfactory center in the animal's brain. And we'll watch how information about smell percolates into the brain and is changed by the animal's ongoing behavior. So that's a very...
So do you also have sensors on the mouse as it's moving around? So you're actually sensing what's going on in its brain?
Oh, yeah, yeah. So also we can implant tiny little miniaturized microscopes that actually let us visualize individual cells. We can implant silicon probes that let us capture the electrical signals that allow neurons to communicate, so-called spikes that the neurons use to talk to each other. And we often record all this information while simultaneously measuring how fast the animal is sniffing or breathing, while simultaneously measuring all of the three-dimensional movements of its body, and while controlling, some sensory object in the world, like a smell. And sometimes we even have techniques that allow us to essentially stimulate specific neurons in the animal's brain to, for example, recall, as we were discussing, a past memory, or to implant into the animal's brain a fictive sense, like a fictive odor. So like we can trick the animal into thinking it's smelling something when actually nothing's really there. We can ask what the consequences of that are. So those are the kinds of techniques we often use.
The one thing I've begun to appreciate working with colleagues at the medical center, I'm working with people at the Rinberg lab, is the kind of engineering actually that goes into being able to collect this kind of data, right? So you were talking about probes, so we have this method where we remove a part of the plate so we can observe the glomerulus. But we're only observing, the dorsal part of it, right? So we're only getting, 10% of the sort of possible neurons we're observing. How how much effort goes into the actual engineering before you are able to collect data? And how frustrating is it to collect this data?
Yeah, so most of the effort goes either to the engineering or to the analysis at the very end. The actual experiments are like some tiny fraction of the overall amount of time.
I know, I have developed a huge new sort of sense of respect for neuroscientists, doing this kind of work, because there's so much cost and effort that just goes into the setup before you even, have observe your first sniff.
Yeah. So just to give you an example of like the kinds of engineering challenges we face in my lab, we do things like build custom cages for animals that have 8 synchronized cameras, 2 of which see in 3D and 6 of which see in 2D, all of which are running at 120 hertz, all of which have to be synchronized. And we run these cameras for months as we videotape a mouse living its whole life, as we have a silicon probe implanted in the brain and we're continuously 24/7 streaming out neural data, as we're offering the animal different odors and different objects to interact with, as we're monitoring its sniffing, as we're manipulating its neural activity. It's a tremendous engineering challenge, and it's a tremendous kind of data handling challenge. We generate data sets that are petabyte scale. Months and months of 120 hertz imaging data, plus electrophysiology, plus other forms of data like sniffing data, combined, finding ways to keep things aligned and synced, making sense of the relationships between these data streams. It's truly a tremendous engineering challenge, and that's half of what we do in the lab, is to deal with those engineering challenges.
Yeah, I can imagine, because, when you were talking about using machine learning methods to actually understand how the mouse is moving around the bucket, I was thinking like, wow, this is a huge amount of data. How much of that involves sort of human input versus stuff being done automatically, right? Because presumably you're using, some supervised machine learning methods. And so how do you code all this up? Is it done mostly automatically, or is there a lot of manual effort going into coding the data?
So actually, I'm glad you brought that up. So we don't use supervised methods nearly ever. So we have adapted algorithms that were designed to parse natural language in an unsupervised way, to parse movements, essentially. And so we built a platform called motion sequencing, or MoSeq, we call it. And MoSeq is just fundamentally, an unsupervised data-driven technique that identifies repeatedly used units of behavior. And it turns out that if we perform simultaneous neural recordings in motor centers as we perform motion sequencing, the units of behavior that motion sequencing discovers, that the machine learning algorithm identifies, those units of behavior have explicit correlates in the brain. And we can make neural manipulations. We can actually change activity.
And so just sorry to interrupt. So when you say units of behavior, what are you talking about? So what's an example of a unit of behavior?
Great. So we refer to each of these units as a behavioral syllable. They're brief, three-dimensional stereotyped motifs of action, things like the first half of a rearing behavior, or a sniff to the left, or a step to the right. They tend to last 300, 400, 500 milliseconds long, although their duration distribution can vary from 100 milliseconds to a couple hundred milliseconds. And they, tumble one after the other in time in a somewhat predictable manner. And so we can look at the brain and actually see evidence that the brain is creating behavior out of these parts and placing these parts into various sequences that allow animals to solve whatever problem they're solving, which typically in my lab is to explore a little bucket and to interact with an odor, or to interact with another mouse, or to play with an object, or to collect food, or to, go get some water, to solve kind of natural ethological problems. And the gratifying thing here is that, as I was mentioning, and I'm sure you're aware, unsupervised machine learning techniques, and even supervised machine learning techniques, can essentially arbitrarily break up data into parts. And you can be kind of convinced that the data has some sort of structure because, that's what your machine learning algorithm under the hood kind of, built in, right? And so we've been very careful to link our analysis of behavior and ground it to actual neural recordings, which has taught us that the particular models we're using are at least in part telling us something that's true. that really mice really do build their behavior out of predictable parts. I think it's probably true for all animals. The parts might exist at different timescales and be organized in different ways, but it's almost certainly true that to some extent all animal behavior is modular. And in our particular case, we can really validate the existence of these modules and sequences by combining the machine learning with actual ground truth neural recordings.
Very, very cool. Just by the way, a peripheral question: do you believe in? What do you think about free will? A lot of debate about free will.
I have. It shouldn't surprise you. I have no opinion about free will except to believe that I have it. Well, I mean, what is true is that, and I think this is the thing in the lab that I'm most excited about, to be honest, is that what we learn from these kinds of methods is that mice of the same species encountering the same problem tend to generate similar patterns of behavior, but their patterns of behavior are highly, highly individualized. So if I take a set of 100 mice, okay, and I run them around in a bucket with approximately 100% accuracy, I can tell you which mouse is which based upon the specific way any individual mouse is just exploring the bucket.
Wow,
and that's crazy.
Cool.
And that's stable. So like the same mouse will exhibit a kind of stereotype pattern of exploration that's stable over timescales of months. We're actually in the process we have a paper under review where we've imaged the behavior of mice over their entire lifespan. And so so we can think about how their patterns of behavior evolve as they move from being juveniles to adults to being old. And one of the key things that pops out is that, mice are truly individuals in a way that we haven't appreciated. And this is super important for biomedicine. In the experiments you're referring to from Dima Rimberg's lab, and basically in almost all experiments we do in biomedicine, we think about these mice and animals we use, fruit flies, worms, as basically widgets, like a generic widgets that are all identical, and we can get some data in a bunch of these widgets, average all the data together, and truth emerges. But these are actual animals that have had their own experiences and their own developmental histories. And those histories are indelible and revealed just by watching 10 minutes of video, which just tells you that they all have their own little personalities that are stable. And it's something, and so I've become increasingly interested on both the sensory side and on the behavioral side in individuality. So, we were talking about smells a little bit earlier. I'm talking about how, you and I share some idea about the ways that smells relate to each other. Again, going back to the example, lemon and orange are similar and different from pizza. So we've done some math, right? So we've done some math where we look at the ways that smells are represented in the cortex of a mouse. And it turns out that about half of that relational map is in common between mice, right? So about half the information is shared, but half of it is individualized and personalized based upon each mouse's unique experience.
So does that mean that if a mouse smells something, you get sort of some part of its response is kind of an average, and something is quite unique?
Yes, exactly. And that explains a little, and we think that relates in a kind of deep way to the question you were asking before about culture, right? I mean, your experience is different from mine, right? And so necessarily the map of space in your brain is different from the map of odor space in your brain is different from the map of odor space in mine. But it's not permanently like that. It's kind of constantly evolving and changing. I'll tell you about an experiment we recently published that I'm super excited about because it's super unexpected, which is that, your nose itself has a kind of map of odor space. It understands relationships between odors, and it encodes those relationships through patterns of activity in the odor receptors themselves. So imagine, let's just go back to my example of lemon and orange. Okay, so lemon and orange smell similar because the small molecules that come off a lemon and the small molecules that come off an orange, and that's what we're smelling, those volatile odors. Those small molecule bouquets have many components in common. There's some unique components that let us tell lemon and orange apart, but there are many, many common components that let us know that these two things are related. So when we take a sniff of lemon or orange into our nose, I told you that your nose has 400 receptors. Some subset of those receptors, maybe 40 or 50 of them, are going to be activated by lemon. and 40 or 50 receptors are going to be activated by orange. And there's going to be a substantial overlap between the set of receptors that are activated for orange and lemon. Right? So your nose itself contains a map of these relationships. The receptors that are going to be activated for pizza are going to be different. And what we've shown recently is that based upon your previous experience with odors, your olfactory sensory neurons in your nose change what they respond to such that those relationships in your nose change. And so your perception of whether lemon and orange is similar or different, that very code in your nose is flexible and changes depending on what you yourself have recently smelled.
Wow, fascinating.
Yeah, so the whole system is plastic. The whole system, from the nose into the brain. I mean, it's really unprecedented how flexible the system is. It's as if it's built to instantiate your everyday experience with smells.
Yeah. So, Sandeep, I want to come back to sort of, I guess, the clinical possibilities with smell. And, I'd referred to Joy Milne earlier, and the fact that she smelled Parkinson's almost 15 years before her husband was diagnosed with it. And that, there are people who say that these many of these illnesses, once you detect them, it's like, it's already over, right? That once you actually, find that someone has Parkinson's or Alzheimer's or, a number of these diseases that develop over a long period of time, it's too late. And then you're treating the symptoms, right? And there don't seem to be any sort of biomarkers that have been identified for these diseases, and yet people like Joy smell them with 100% accuracy, right? Now, she's hyperosmic. By the way, are you hyperosmic?
I'm not. I actually have a relatively poor sense of smell, although I enjoy smelling things. My wife will tell you how poor my sense of smell is.
That's so ironic. But coming back to Joy, so what is it that's going on there that's enabling her to do this? And what will it take to make this process digital, right? So that computers can actually start doing this?
I'm going to actually give you a realistic answer to your question. So normally as an olfactologist like myself, the answer that I would give you is something along the lines of, humans are capable of smelling parts per trillion of odors. So we actually have very sensitive noses, and, hyperosmics like Joy Milne, might be able to do even better than that. And so the fact that, she can smell Parkinson's suggests that if we could somehow screen through some sort of chemical detector, we would be able to detect Parkinson's and Alzheimer's and other diseases early. So I think that is possibly true, but I will say that our technical ability to search for biomarkers in the blood is increasing by leaps and bounds. And, those of us who, my lab has some interest in Alzheimer's disease, those of us who are kind of familiar with the space are really seeing the evidence that our ability to detect things in the blood, in the plasma, in the CSF today is nothing like it was 5 years ago. And the trajectory is kind of exponentially upwards. And so I think many of us think that Joy Milne really is a kind of proof of concept. She tells us that there are biomarkers that are detectable very early. And they exist in humans. And so we should be able to find them. It's not clear whether the way that we're going to find them is through smell. It could be that we find those biomarkers through blood or through plasma or through cerebrospinal fluid. But it's clear that those biomarkers exist. So, I view this kind of anecdote as evidence that we should be continuing to work hard at biomarker discovery. And I think, thinking about ways in which we can maybe, use smell as a new kind of biomarker, I think that's an important avenue. But my suspicion is, my gut suspicion is that the technologies that we have in hand for understanding what's in the plasma are getting so good that actually, within a few years, we probably will have a much richer set of biomarkers to play with than we do today.
So that's interesting. So you say it's a proof of concept that there are biomarkers because of the fact that this is being secreted through sebaceous cells.
Yeah, so we should be able to find
shows
something.
Up.
Yeah,
right. So there's some sort of underlying biology that's
we just
secreting this, and you're discovering it. So there's got to be a biomarker.
Exactly. We just haven't accessed it yet. And I think there are technologies that probably not related to smell that will get us there. Yeah. So I think there's a lot of excitement about electronic noses, for example, like using artificial sensors to detect odors, to quantify them. I will say that in my limited understanding of that space, those technologies right now are still pretty crude. That our ability to detect odors in the broad sense, like to be able to, say, take the scent of lemon and to somehow digitize it into some set of information about chemistry that we can then describe. I mean, we obviously have technologies to do that, but they're not scalable in the way that you want for screening humans, for example.
Right. By the way, again, slight tangent here. One of my previous guests, Alex Wilczko, was he a former student of yours?
My former graduate student.
Your former graduate student,
indeed.
yeah. So, I went to a fascinating talk that he gave, and then I had him as a guest, and he's now the CEO of Osmo. .ai. So just for the listeners, if I hope I'm describing this correctly, essentially what they are able to do is predict odor purely from molecular structure.
That's right. They can predict some aspects of odor quality, whether it's going to smell, a particular bit of chemistry is going to smell like pineapple or mango or smoke, just by looking at the chemistry itself, which is incredibly powerful.
Yeah. So how far will that technique take us towards the clinical problems of diagnosing these diseases that develop over a long period of time?
Yeah, so,
is that
So I think these techniques. Just full disclosure, I'm an advisor to that company, Osmo. So I don't want any of your listeners to think I'm saying something out of school here. Yeah, so that technique, the use of machine learning to look at chemistry and make predictions about what things are going to smell like, is incredibly important and powerful, right? And I want to remind you that what it's essentially doing is tapping in to those aspects of odor perception that are common between me and you, right? By necessity, those algorithms work on the average across people. And so they can't tap into what's personal to me or to you, by definition, right? And so the limits of those algorithms are precisely in those spaces where smell is most personalized. And the strengths of those algorithms are precisely in those spaces where, odors are shared in terms of their perceptual qualities. And honestly, as humans, we don't know the boundaries. We don't understand quite the degree to which our odor space is truly shared versus truly personalized. I think you and I both have some intuition that it's a bit of both. And I want to just mention one important quirk of anatomy that maybe you don't know that I think is really relevant here, which is that information that you smell in your nose gets sent to a structure called the olfactory bulb. And then in parallel, it goes to centers in your brain responsible for olfactory processing. It goes to centers responsible for learning and memory. And it goes to centers responsible for emotion. And that is very different from all the other senses, which have to hop, from station to station to station before they arrive at a place in your brain involved in memory or emotion. And so your smell system is really hardwired for emotional responses. It's designed for you to have feelings about what you're smelling at any given moment in time. And you might imagine that that's very personal, right? And so it's not clear how well these machine learning algorithms will marginalize away the personal part and really capture the shared part. And I think a debate in the field is how important is that? For each one of our smells, our senses of smell. All that being said, right, there are perfumers who make perfumes that all of us like, right? And so it's clear that some huge amount of smell perception is in common. And as a consequence, these machine learning algorithms are going to be incredibly powerful. And I do think that these kinds of algorithms that allow us to go from chemistry to perception are going to be important for understanding and digitizing smell ultimately, right? For understanding how chemistry is transformed into perception, which is ultimately required if we're going to figure out ways to simplify, for example, the transmission of information about smells. So just to remind you, you and I are looking at each other over a video screen. And that's because we understand the basic ways that light can recombine to give different colors, right? And so it's not like there's like some painter in my video screen that's constantly painting with 1,000 different colors. There's a basis set of red, green, and blue that can be combined in various ways to give you all the colors that you can see. And that's how this video screen works. And that's how I can see you in full color and you can see me in full color. And we don't have a sense of what RGB is for smell. So I think the thought is that these machine learning algorithms that transform chemistry into perception might help us understand the equivalent of red, green, and blue for smell. And if we can understand that, we might be able to build machines that transmit smells between me and you at a distance, which is something that we don't have now.
What will this RGB of smell look like, right? Because with vision, we have this external stimulus. It's a purely physical kind of thing. But with smell, there's this sort of, there's the molecule, and then there's a chemical reaction going on, and there's a transmission of information. So what will these primitives, these RGBs, look like for smell?
I think that's a good question. We don't really know, I would say, but I can tell you what the dream would be. The dream would be that there is some small set of odors that we can identify, odor chemicals that we can identify. Maybe it's 10 of them, maybe it's 50 of them, that when combined in various concentrations, effectively recapitulate every possible perceivable smell. That'd be the dream. And then you could have a machine with just these 10 chemicals and, a computer that could mix them in the proper ratios that could then create any smell you want. Now, whether that is an achievable thing, whether that's possible at all, is not clear, because we don't know what those 10 odors or those 50 odors should be.
Does that seem like a plausible kind of theory to you, that there would be like a really small underlying set of primitives and then concentrations do the trick?
I think there is quite a bit of debate about whether or not smell is fundamentally low dimensional. I think many people believe that it is. That is, that there are just a limited number of axes along which smell is organized. You can imagine one axis is pleasant versus unpleasant. You can imagine a couple other quality-related axes, right? And that maybe if you can organize a low-dimensional space out of smells, and you could find smells that drive you along those axes, you could actually build a basic set of odors that you could use for digital olfaction.
So where do you fall on that? Because I've heard you refer to smell as being high dimensional.
That's right. I am undecided. I do a lot of So I do a lot of recordings of neural activity in the brain, where I see all of the richness and the high dimensionality of what smell can do. So, the perspective that I bring to this is one in which, you give a smell to a nose, and all sorts of incredibly complicated patterns of neural activity emerge. Right? It doesn't look simple. It looks very, very complicated. Right? And so, from that perspective, it feels very high dimensional. Our human intuition about smells is that it's pretty low dimensional, right? That there's just a few kind of common categories of smell that we can discuss, we talk about with each other, and all smells can be described as some combination of smoky and citrusy and floral and cheesy. And if we just had enough of these categories, we'd be good, right? And the truth is probably somewhere in between. But for me, I would love to get a handle on if smell is really low dimensional, how the brain does that. How does the brain take, for example,
or maybe even if it is high dimensional, how does it reduce it to this dimensional space, right?
Exactly.
Because this has a temporal element to it too, right?
Absolutely.
Smell also is temporal.
That's right.
Yeah.
And so understanding where low dimensionality emerges. Is it a property of something that's happening in the nose? Is it something the brain is imposing on smell? A related question, which I think is super interesting, is fundamentally, is the way that a mouse brain organizes smells the same way as our brain organizes smells, is the same as a fly brain? Are they similar or not? So you're referring to this machine learning work that Osmo and Alex Wilchko did and published, where they could take individual chemicals and predict what they're perceived like. The machine learning algorithms they developed work equally well in fruit flies, in mice, and in humans, arguing that we all share a grand perceptual space across evolution. That's an argument. On the other hand, I told you that there are more olfactory receptor genes than any other gene type in your genome. They're also the fastest evolving family of genes. So the receptors for smell are being sculpted by evolution to allow each species to specifically interact with those smells that are most important in its ecological niche. That argues that smell should be very specific to different species, right? And the nature of smell should differ from a fruit fly to a mouse to a human.
Right, depending on the environment.
Exactly. And the niche. And so all these things are true all at once. And I think, we have to sort out, the degree to which smell is a fundamentally shared thing versus a fundamentally personal thing, whether it's, specific to individual species and organized in a species specific way, or really fundamentally shared across all organisms. And there are good arguments to be made on all sides.
One of the things that I started to think about when reading your work was like, what does meaning actually mean, right? Because like, is meaning something that's inherently low dimensional, right? Because,
Yes.
do we ascribe meaning to things only because we're able to describe it like in a simple way? Otherwise, we don't understand what something means.
Yeah. I think it's a wonderful question. And I think it does tie very nicely to this question of low dimensionality. So, in the most primitive animal, right, a single-celled organism. Imagine a single-celled organism in a undifferentiated sea just floating around, right? The basic thing that cell has to do is distinguish things it wants to approach from things it needs to avoid, right? It's just a single cell. It can only go forward or backwards, basically, statistically, right? It can do one of two things. It can go towards a thing or away from a thing. That's very low dimensional. Even though there are many things in the environment that it might have to interact with, ultimately, there are only two behavioral outputs, right? Good or bad. And there are arguments that have been made by folks like Paul Sisek at the University of Montreal that, that's actually how our brains initially were organized. We had like some basic low dimensional axes where we just kind of put different stimuli in the world into buckets, low dimensional buckets. This is good, this is bad. And evolution hardwired into our brain the meaning of different sensory cues. Whenever you smell X, it's good. Whenever you smell Y, it's bad. And the whole brain was built on top of that low dimensional system. That idea seems very appealing to me.
Fascinating. Really fascinating. So Sandeep, we started the conversation and with, how you got into this space. You said you wanted to be a banker and do economics, and good thing you didn't. And I have listeners, who listen to the show. Some of them are, even high school students. What would your advice be to young people who are interested in smell and the brain? Is this a great time to get in? Is this the ground floor? Like, how would you advise people to think about this?
This is an amazing time for neuroscience in general and for olfactory neuroscience in specific. The sorts of tools and technologies we have access to now were unimaginable a decade ago, essentially. The ability to read and write information into the brain of a living, behaving mouse as it freely interacts with the world. That was just unthinkable, right? 10 years ago, 15 years ago. And we also have just a host of molecular biology tools that doesn't just allow us to look at how neurons talk to each other, but to actually look at the innards of the neurons, the genes and the proteins that make up neurons, and to ask how they themselves change as a function of experience and time. And so for me, I'm like a kid in a candy shop. I wish I were 20 right now because there's so much to discover. I hope from this conversation you get a sense that like there's very little we actually know about olfaction. It's just this deep mystery, right? You have a cup of coffee in front of you. You take a sniff. You're sniffing in 800 separate chemicals, and somehow your brain turns that into this incredibly appealing singular percept. How does that happen? That's the essence of perception. We don't know anything about it, I argue. And so it's really a time where the neurophysiological techniques and the mathematical techniques and the computational techniques are coming together in a way that allows us to respect and understand the biology. And so I think it's just a tremendously exciting time, both for neuroscience and for olfactory science in specific.
Very cool. I tell people that this is also, people get into AI. I tell them that I'm envious about the level at which they're starting from, because when I got into the field, the tools were so primitive. Most of the current tools didn't exist then, and people are just starting from a whole new place. It seems like all of these things, the biology and the computation, the state of the art is just progressing at an amazing pace. Well, Sandeep, this has been fascinating. Thank you so much for your time. I learned a lot by reading your stuff, and I learned even more from this conversation, and I really enjoyed it. Thanks. I really appreciate you coming on the show.
Thank you so much for having me. I had a great time.